Okay, so hello, my name is Risong, and my project is Mendel's lentils, identification and analysis of the zero-tannin gene in lentil. Lentils are an important global pulse crop, providing an essential source of vitamins, minerals, and protein to many people worldwide, especially those in developing nations. Canada is the world's largest producer and exporter of lentil, producing nearly 1/3 of the world's supply. Most lentils have lightly colored flowers, reddish stems, and dark seed coats, as you can see in figures 1 and 2. Now, this pigmentation is caused by condensed tannins, or CT, and anthocyanins. These are present in the polyphenolic pathway. Now, the same CT that are responsible for seed coat pigmentation are also anti-nutritional. This means that they bind to iron, proteins, and minerals, reducing their availability to the human digestive system. Now, there is a commercial zero-tannin or ZT lentil variety available with clear seed coats, as you can see in figure 4. Compared to pigmented varieties, the ZT lentil variety may have reduced anti-nutritional qualities. However, it also has poor disease resistance and low yield, possibly due to its lack of CT or other biochemical compounds associated with general disease resistance. These qualities have severely limited the commercial potential of the ZT lentil and therefore its availability to consumers around the world. Now, the key to improving the commercial potential of the ZT lentil may lie in its more resistant relative, the commercial pea, shown in figure 6. Most commercial peas are actually zero-tannin and yet have adequate resistance and yield, contributing to their commercial success. The PZT gene is Mendel's A gene, which is a transcription factor in the polyphenolic pathway, which is also responsible for purple versus white flower color in pea. Now, it is highly likely that the lentil homologue of the PZT gene is responsible for the lentil ZT trait, as pea and lentil are closely related and the same genes are often present in both species. Now, the mystery becomes, if the same gene is responsible for the ZT trait in both species, then why are ZT peas resistant while ZT lentils are not? Is this due to linked poor resistance genes, or polymorphisms in the ZT genes, or other genes in the polyphenolic pathway? The first objective of this project was to confirm the lentil ZT gene's similarity to the PA gene by developing markers for the lentil ZT trait based on previous genetic information from P. The second objective was to determine differences in gene expression patterns in the polyphenolic pathway between ZT and pigmented peas and lentils. So, one known difference between ZTP and ZT lentil is that artificial selection for the ZTP began approximately 700 years ago, long before such selection in lentil. And this long-term breeding may have reduced the linkage between poor resistance genes and the ZT gene. The reduced linkage may be the reason for the differing resistances instead of polymorphisms in genes between both species. Therefore, I hypothesize that the ZT gene and its expression in lentil and pea will be similar. So, the genotypes used in Objective 1 were LR30, a lentil recombinant inbred line population, which is a cross between CDC robin, pigmented, and 2670B, a ZT line. It segregated for the ZT trait, as well as the lentil diversity panel, or LDP, a group of commercial pigmented cultivars. So, samples were grown up, and DNA was extracted with a micro-CTAB extraction procedure from young leaf tissue. Caspar primers were then designed for 2 SNPs from the lentil homolog of the PA gene from previous 454 sequencing data. Using the DNA as the template and the Cas-bar primers as the assay, Cas-SNP genotyping was used to screen the LR30 population and the LDP. The resulting SNP allele data was then correlated with the observed phenotypes for each sample to determine the accuracy of the marker. So the results of the Cas-SNP genotyping indicated that of the 2 primers designed, only one, LCC 336, was polymorphic between the parents of the population. When it was screened on LR30, it co-segregated with the ZT trait. As you can see in figure 11, there is distinct separation between the ZT allele group, shown in blue, and the pigmented allele group, shown in green. When this was correlated with the phenotypes for each sample, the SNP alleles and phenotypes matched. And the LR30's behavior, the SNP's behavior on LR30 indicates that there is extremely likely that the lentil and PZT genes are similar. Now, when it was screened on the LDP, the results were scattered. Now, since the LDP is a group of pigmented cultivars, it is expected that all samples display the pigmented SNP allele, similar to the green group in the LR30 screening. As you can see, there are many heterogeneous samples and pigmented samples with the supposedly ZT SNP allele. And this supports the theory that the lentil ZT mutation is indeed not a SNP, but may be a deletion similar to the PZT mutation. So moving on to objective 2. The genotypes used for the gene expression studies with the lentils CDC Gold, ZT, TMP 9586, ZT, and CDC Robin, which is a pigmented line, as well as the P's CDC Bronco, ZT, and CDC Rocket, a pigmented line. So samples were grown up, and root, shoot, seed, pod, and flower tissue were collected and immediately submerged in liquid nitrogen to prevent RNA from degrading. RNA was then isolated with the following 4 main steps: lyse, filter, bind, and dilute. Primers were then designed around select genes downstream of the A gene. Now, the A gene is the homolog of the Arabidopsis TT8 gene. LDOX, or leucoanthocyanidin deoxygenase, is responsible for anthocyanin production, which is involved in flower coloration. Banules is involved in CT production, which accumulates in roots and seed coats. Elongation factor 1 was the control, as it was expected to be similarly expressed across all genotypes. So, using the sample RNA as the template and the design primers as the assay, reverse transcription real-time PCR was conducted. The delta-delta CT method was used to convert the raw RT-PCR data into relative gene expression figures, with all expression levels being relative to the CDC Bronco sample. So, currently there has only been one biological replication on one technical replication. Thus, no definite statements can be made. However, the general trend of the gene expression studies can be observed. A gene expression was actually expected to be similar across all genotypes because the primers were designed outside of the polymorphism, but still in the gene itself. Now, as you can see, A gene expression was actually variable among all genotypes, and this will be confirmed with further replications. Banules is involved in CT production and is expected to be higher in pigmented varieties, shown in pink, than in ZT varieties, shown in green. In the seed data, the lentils actually reverse the expected trend, with the pigmented lentil expressing less than the ZT lentil. Another tissue of interest is the roots. While the two Ps had very similar expression levels, the pigmented lentil expressed much more than the ZT lentil. Now, this is interesting because BioGENeius leads to CT production in roots, which is associated with resistance against soil-borne pathogens. LDOX is involved in anthocyanin production for flower color, and it was expected to behave like BioGENeius. The flower data supports this, with both the pigmented lentil and P varieties expressing more than their ZT counterparts. However, again in the seed data, the lentils reverse the expected trend, with the ZT lentil expressing more than the pigmented lentil. So, what does all this that I've told you actually mean? For objective 1, the Cas/SNP genotyping, particularly the LR30 screening, increases the likelihood that the same gene is responsible for the ZT trait. The scattered results from the LDP screening indicate that the SNP is not causative and functions as a marker via linkage. A potential explanation for the marker's differing behavior on LR30 and the LDP is that the SNP arose first, and then the ZT mutation arose in only one individual with one of the SNP alleles. This would explain why there are some pigmented individuals with the supposedly ZT SNP allele. So the general trend of the gene expression studies indicates that lentil and P data do differ, particularly for LDOX and BioGENeius expression in roots and seeds, two tissues that are associated with resistance against soil-borne pathogens and general disease resistance. Now, while the hypothesis predicted that expression levels would be similar, the differing trends introduce the possibility that polymorphisms in the ZT genes or in genes in the polyphenolic pathway are responsible for the differing resistances. Future studies will include conducting more biological and technical replications of the gene expression studies, sequencing the lentil ZT gene, and using bioballistics to confirm it, as well as analyzing the biochemical properties of the lentil ZT gene and comparing it to those of the PA gene. More genes up and downstream of the A gene will also be tested to gain a better understanding of the A gene's effect as a transcription factor in the pathway. Ultimately, an understanding of the genetic differences between ZTP and ZT lentil that contribute to the P's enhanced resistance will aid in increasing the commercial potential of the ZT lentil. Not only will this open up new markets for producers, but also provide consumers around the world with increased access to the nutritional benefits of the ZT lentil. The reduced anti-nutritional qualities of the ZT lentil also make it an ideal vessel for biofortification of essential micronutrients. As well, increased insight into the polyphenolic pathway in pulses will aid plant breeders, as the pathway is associated with pigmentation, taste, disease resistance, and yield. Many thanks to my mentors, Dr. Kirsten Bett and Mr. Rob Stonehouse, for their support, help, and guidance throughout the project. As well, thank you to Dr. Ravindra Chabar for reviewing my presentation. Thank you to Dr. Puba Ganeshan for helping me with gene expression analysis. And the University of Saskatchewan is greatly appreciated for their continuing support of my research. As well, thank you to Wordmark Consulting Incorporated and Sanofi BioGENEius Challenge Canada for making my participation in the challenge possible. Thank you for your time. Do you have any questions?
Thank you, Ree. Questions? Ron?
A couple of things, a couple of technical things, but before we get into that, we heard at the beginning that you were involved and were in fact a winner of this competition 2 years ago. I wasn't a judge 2 years ago, so can you tell us is this a different project completely, or is this ongoing work from what you've done over the many years you've been involved with this competition?
My project in 2010 was involved with a lentil pathogen, and I developed molecular markers to identify different races of this pathogen. This year I'm working with the plant itself, still working with lentils because I believe it's extremely important as a resident of Saskatchewan that produces 99% of Canada's lentil. It's really something that I can have a huge impact right in my own backyard. But this indeed is a very different project, still associated with lentils, but it's a different area.
Okay, and a technical question. For your gene expression analysis, you designed primers and you showed us the scheme. How were those primers designed? Were they manually designed based on the sequence, or did you use some kind of a freeware program, or how did you go about that?
Those primers were actually designed manually with sequences from either the current lentil database from the University of Saskatchewan or NCBI.
Okay, and just finally, you talked about using bioballistics to confirm the sequence. Can you sort of explain what that means? I'm not quite sure I understand the continuum between sequencing a gene and then using bioballistics to confirm the sequence.
Okay, so essentially what that would mean is that the gene would be sequenced, and then bioballistics would be used to make a ZT lentil flower, basically produce anthocyanins for that flower color. So that sort of confirms the gene that is responsible for the purple versus white flower color. Does that answer your question? Thank you.
Yes, you mentioned that you use delta-delta C2 method for data analysis. Can you tell us a little bit about that?
Okay, so what the delta-delta CT method is CT is the cycle where the expression crosses a preset threshold. And this threshold was set in the log form of just the raw RT-PCR data curves. And what delta-delta CT measures is it measures this change in CT relative to first your endogenous control, which is your EF1, elongation factor 1. And everything is also relative to the CDC broncho samples, which, as you'll see in the expression figures, has a relative expression of 1, with everything else being relative to that sample.
So yield is an issue with the ZT lentil, and that's number one for producers in Saskatchewan. Are you proposing that the gene deletion could be done transgenically, so to create a transgenic lentil?
Okay. The thing with transgenics is very iffy, especially in crops. So essentially what we'd be proposing to do is set up a breeding population with the most resistant lentil variety that is out there and the ZT lentil variety, and continuously breed them until you get an individual with the ZT trait as well as enhanced resistance.
So the tannins are presumably used in the structure of the plant as well, right? So are you going to see any, would you predict seeing any changes in the standability of the crop?
Well, tannins have a variety of functions in plants. Disease resistance as well as taste. They give plants like sort of astringent taste. They're also used, because of their astringent taste, they're also used as an effective defense against things that would eat lentils. In terms of the other aspects that would be affected, just from growing them at the field house and at the greenhouse, we see that they're extremely prone to insect infestations. They generally have trouble germinating. That's because there's no tannins in the seed coat itself, so there's very little protection against soil-borne pathogens. Does that answer your question?
Okay.
I'm just curious about the turnaround time for all these experiments. So, and that's that partially once you said you had to go back and confirm a lot of these.
Mhm.
So I don't know anything about lentils except what they taste like, so I can't really speak. But in terms of growth, I mean, it goes back a little bit to what you were saying, but in terms of getting down the road where this is going to be commercially viable to the lentil manufacturers, you know, what is that process overall? Like, how long does it take to do this whole set of experiments? If you weren't just doing it part-time because you're going to school.
Okay, so I had initially set out to actually sequence the lentil ZT gene, but the gene is about 3.5 kilobases, so that was, we had a lot of trouble like amplifying that. That took, like just growing up the population took a lot of July to August, so like that was full-time work because I was off in the summer. As for the gene expression studies, we've actually been having trouble germinating the lentils and the peas. For some reason, bacterial and fungal infection has just been getting at them really rapidly. But in terms of a timeline, let's see, I've been doing the gene expression study. I grew them up mid-December. And by about late January to early February, they were ready, and I conducted gene expression in the early days of February. So just that.
Okay, just quickly, can you, the quantitative RT-PCR, the RT-qPCR, were you able to use the instrument yourself and do all of that experimentally? Or it's fairly complex instrumentation, and I'm just wondering if you were able to do those particular experiments completely on your own.
Are you referring to the RT-PCR
Yes.
or RT-PCR?
RT-qPCR.
Okay. For those experiments, actually, we had like many replications. So, like, you know, in the very first set of experimentation, I was helped along by my supervisors. But as I kept doing more and more repetitions, I became increasingly more independent with using the machinery and using the various assays and etc.
Any other question? Ari, talk to us how much time you spent doing this during this whole period of this part of the competition.
This part of the competition.
I know you've been around doing this work in different ways, shapes, or forms for a long time. Tell us about this. Give us a rough idea.
I started work on the project just growing up samples mid-July. So mid-July to the end of August was basically spent in the greenhouse growing up samples, extracting DNA. And then I just worked continuously throughout the school year until actually around late March. And I tried to start up a new wave of gene expression studies. So I've been working on that really up to the competition. But like I said, the germination just didn't work out and the timing was just off.
So how many hours would you roughly guess you spent on doing this?
You know, on average, like some weeks I just wasn't able to devote that time. But on weeks that I had off, I would spend maybe 8 hours a day at the lab. So I'd say like an average of 10 hours per week. Yeah.
Okay. Let me just ask you a slightly different question. If in fact you had to, I mean, this whole controversy about genetically modified foods, not being able to export it to Europe, India, of course, a great market for Saskatchewan, etc. How would you address some of those questions to the lay public?
Okay. Well, I really think that when you're talking about like ideal crops, it's really what the consumer wants. Like regardless of what the researcher thinks is best, it's the consumer that's going to be eating these crops, the consumer who's going to be buying those crops, and you always have to think of what the consumer wants. And even though what the consumer wants might not be logical, might not be like seem to scientists, but it always depends on the consumer. And I think that in terms of like genetically modified foods, there has to be that communication with the public. You have to, make them aware of what's like the actual scientific process behind these. There's actually at my regional competition, there was an interesting story going around. Like there was a survey sent out about genetically modified foods and whether people would eat them, and some people answered no, and I mean that's perfectly fine. That's within their belief. But when they were asked the reason for it, the reason was because there's DNA in the food. Now, when you have that kind of reason, it sort of becomes that, it's sort of like an alarm, because If people are scared of genetically modified foods because there's DNA in the food, then that shows that there's a disconnect between the public and the researchers, and that needs to happen in order for genetically modified foods to become viable.
Thank you very much for coming back, and good luck.
Thank you.
Enjoy Ottawa for the next 24 hours.
Thank you.